Epistasis is where gene interaction leads to a gene blocking another gene's effect. Let's talk about single dominant epistasis and shoe color.
3 shoe color possibilities: white, tan, and black. So we can go from white precursor to tan intermediate to the end product black. Gene A produces an inhibitor that blocks enzyme 1.
This causes the shoe to be stuck with white. However, if there are too little A's and therefore an inhibitor A is not present, then this allows the shoe to be tan. And if a shoe possesses too little A's and a big B, this allows the shoe to be black. However, if the shoe has too little A's and too little B's, therefore a big B is not present, the shoe is stuck being tan. Next, we're going to look at the proportion of phenotypes of a dihybrid cross.
First, we're going to set up our 9:3:1 ratio with our genotypes.
In order to figure out the phenotypes of the genotypes, we're going to take a look at our epistasis pathway.
Our first genotype has a big A and a big B. The big A is going to produce an inhibitor that is going to block the enzyme 1, which is going to make the phenotype white. Next genotype is a big A and 2 little B's. Again, there's a big A that produces an inhibitor that blocks the enzyme 1 and keeps the shoe being white. Next genotype has 2 little A's and a big B. Because of the 2 little A's, the shoe gets to tan, and because it has a big B, the shoe is black. Our last genotype is 2 little A's and 2 little B's. Because there are 2 little A's, the shoe gets to tan, and because there are 2 little B's and there is no big B present, the shoe has to stay at tan. So it is tan. Our phenotypic ratio for a dihybrid cross is 12 white, 3 black, 1 tan.
Next, we're going to cross big A, little b, with little a, big B, little b, and find the phenotypic ratio.
Here's the genotypic ratio of this cross, and again, we will be using the epistasis pathway to find the phenotypes of each genotype. So our first genotype is big A, big B, little b, and because there is a big A, and it produces an inhibitor which blocks enzyme 1, the phenotype will be white. Our next genotype is big A, little b, and again, we have that big A which produces the inhibitor which blocks enzyme 1, which keeps the phenotype white. Our next phenotype is little a, big B, little b, and because there's 2 little a's and no inhibitor at big A, we get to tan, and because there's a big B, we proceed to the color black, and the phenotype is black. Last genotype is little b, little b. Because there's 2 little a's and no inhibitor big A, we get to tan, but because there's no big B, we only have 2 little b's, we're going to stay at tan. The phenotypic ratio is 8 white, 4 black, 4 tan.
And here are my references.